Okay, good afternoon, everybody, and welcome to this recorded class, which replaces the class that I missed yesterday. I'm still struggling a little bit, so you'll forgive me if every now and again I break off and have a little coughing fit. But we are going to proceed with our discussion about the proteobacteria. We discussed the alpha proteobacteria last time. And now we're going to discuss another group called the beta proteobacteria, and we'll begin our discussion about the largest group of proteobacteria of all, which are the gamma proteobacteria, which include many of the bacteria that you have already encountered in lab, probably. Okay, so the beta proteobacteria, in common with all proteobacteria, they are gram-negative. They vary in their relationship to oxygen. Some are facultative anaerobes, others are obligate aerobes. I cannot think of any obligate anaerobes in the beta proteobacteria. In any case, some of them will just be mentioned, and this is one of them. This is Acidothiobacillus, also known as Thiobacillus. And Thiobacillus, I'm mentioning this because I'm hoping next week to be able to discuss global biogeochemical cycling of elements. And this is one type which is extremely important to discuss. And these are, Acidothiobacillus is a chemoautotrophic organism. That is, it fixes carbon dioxide, takes carbon dioxide and water, and builds biological molecules out of it in much the same way as would happen in photosynthesis, except that in chemoautotrophic processes, the energy needed to build those molecules is derived from already existing chemicals. And in the case of Thiobacillus, it in fact oxidizes hydrogen sulfide. So have a look at this. Remembering the formula for photosynthesis, right, is CO2 plus H2O and goes to form C6H12O6, which is glucose. So there was H2O. Why was H2O used? The byproduct of photosynthesis is oxygen, right? Because organisms take those hydrogens, take the H2 from H2O, and they use that hydrogen for its reducing power. This is exactly what this organism is doing. It's using H2S, and it is oxidizing it. Is oxidizing it to sulfate, but in doing so is stripping off these hydrogens here and is using them for reducing power. So just keep this in mind, because we will talk about these processes again when we talk about biogeochemical cycling. So the next organism, completely different to Thiobacillus, is Spirulinum. And Spirulinum is a very large bacterium, in fact, and you may well have seen it in lab. As its name implies, it is a spiral bacterium, and it has polar flagellation. Now, all of these features that I've mentioned are important because, first, because they are all distinguishing features which enable us to discriminate between Spirulinum and Spirochetes, which is an entirely different group of bacteria. Spirulinum is a large cell. It is spiral, but it is motile by these flagella at the end. It is polarly flagellated. It usually has a tuft of a flagella, and is found in fresh water. It's obligate aerobe, and it is a very common aquatic and moist soil bacterium. The spirochetes are quite different. The spirochetes are very, very thin, tightly spiraled. They're not loosely spiraled like this. They're tightly spiraled, and they have a flagellum that runs through the center of the cell that causes the entire cell to spiral. And its movement is produced by that spiral movement through the water, like a propeller. A spirillum is completely different. Okay, so the next one that I want to mention is called Burkholderia. And Burkholderia is a remarkable organism because it is able to degrade very complex organic molecules. More than 100 organic molecules are recorded as being degraded by Burkholderia. This has some important implications. The first is that Burkholderia is very commonly occurs in hospital settings. And unfortunately, it colonizes things like polyethylene tubing, all of these kind of artificial substrates can be colonized and degraded by Burkholderia. And in doing so, it often acts as a nosocomial infectious agent. That a nosocomial infection is one acquired from a healthcare setting. And Burkholderia is one of the organisms which can colonize things like catheters very easily. But I mention it not only because of the health implications, but because Burkholderia, its ability to degrade organic matter, and even very, very complex organic matter, even synthetic organic matter, like plastics and those sorts of things, makes it a great candidate for bioremediation, for breaking down pollutants. And there is a tremendous amount of interest in the genes of Burkholderia, in that they're producing enzymes which are very powerful and very unusual. So Burkholderia is of great interest in bioengineering and in bioremediation especially. So the next one is of the basic proteobacteria is Bordetella. Bordetella is a quite dangerous bacterium, especially in children. And it causes the disease of whooping cough. At least one species of it, Bordetella Pertussis, is the one of main interest here. And Bordetella Pertussis, it's obviously gram negative, like the proteobacteria. It's a plump rod. Here you can see it here. These are these red cells here of Bordetella. And Bordetella produces a capsule around the cell, which enables it to adhere to the cilia and to the cells of the respiratory tract. And in doing so, it produces an exotoxin. Actually, it produces quite a lot of exotoxins, but the main ones actually, first of all, destroy the ciliation on the cells of the respiratory tract, and then also begin to kill off the cells themselves. And they shut down that ciliary escalator that moves sheets of mucus from the lungs all the way up to the top of the throat, which is what largely keeps us free of bacteria and particles in the lower parts of our respiratory tract. So in children especially, this produces a very distinctive cough. It's called a paroxysmal cough. That is, you cough, the child will cough and cough and cough and cough until it has expelled all of the air from the lungs. And they will hunch over as the last of the air goes, and then they'll trying to get a breath. They will make a strange whooping sound, like that as they draw breath. The child can become absolutely exhausted, and is extremely debilitating and has a fairly high mortality rate. And so the cells also produce toxins, which can enter the body and cause a systemic infection as well, a systemic response. Usually associated with a very high fever, and it used to be rather dangerous, but there is a very good vaccine for it, and it also responds to antibiotics. So nowadays it is of lesser importance than it used to be, but it used to be a major source of infant mortality, infant and early childhood mortality. So it's Bordetella Pertussis. So the next one you will have heard of, and this is Niaceria. I have mentioned it before. Now this is interesting because it's a coccus. There are not many of these proteobacteria that even get close to being a coccus, but Niaceria is distinctly coccoid. And it usually occurs in 2 like this. But remember, it's gram-negative. So it's one of the very few gram-negative cocci that you will see. There are 2 of particular importance. There are a number of species, but there are 2 of particular importance in healthcare. And these are Neisseria Gonorrhea, which is the causative agent of the disease of gonorrhea, and Niaceria Meningitis, which causes an extremely dangerous meningitis, infection of the coverings of the brain, and then ultimately an encephalitis. Infection of the brain, inflammation of the brain itself, which is extremely dangerous. This is called meningococcal meningitis. There are a couple of other bacteria that will do a similar thing, but meningococcal meningitis is the most common. And so these 2 diseases between them are very, very significant in human systems. So here's Niacera Gonorrhea. Here's a gram stain of it. You can see it's gram-negative, red, and in these little duplicate, little 2s, the diplococcoid form. So gonorrhea, it was one of the diseases which we really regarded as being on its way out. It seemed for many years that gonorrhea infection rates were stable or declining in response, not only to sex education and things like that, but also in response to antibiotic usage. But in about 2015, the number of cases, the incidence began to rise. It rose from 130 cases per 100,000 people, and all the way up to now 188.4 cases per 100,000 people. Let's see, I've put a wrong 0 in there. So it's 188.4 cases per 100,000. And there is grave concern about this. This is attributable to several things. First of all, definitely to resistant strains of Neisseria arising, is normally treatable with antibiotics, but also to the failure of people who are sexually active to observe safe sex practices. And in the early 2000s, from the 1990s and early 2000s, people were very aware of using safer sex practices because of the AIDS epidemic. There was an enormous amount of public information out about how to practice sex safely. And that prevented AIDS infection, HIV infection, but it also prevented other sexually transmitted diseases, infections from occurring, and especially through the use of condoms. But those have become less and less and less well publicized, those methods, and less popular. And this is one of the effects. We begin to see this increase in sexually transmitted infections that are occurring. So Neisseria Gonorrhea, it infects the epithelial cells of the genital tract. Initially, it just infects epithelial cells in the lower part of the genital tract. But it may, after time, it may spread, and it may spread widely. So, for example, a vaginal infection with gonorrhea can spread through the uterus and through the fallopian tubes, and is a major cause of what's referred to as pelvic inflammatory disease, which can actually scar the fallopian tubes, for example, and have very serious consequences. In males, it can spread from the epithelial linings of the penis throughout the reproductive tract, and cause prostatitis, and then even infection of the testes. Here is our cells adhering to epithelial cells, but they also spread between the epithelial cells. Epithelial cells are attached to one another, obviously, and especially in cuboid epithelium, the bacterium can spread down through in between those cells and spread. In females, it causes an initial vaginal infection, might be recorded because it causes a pussy discharge, and also causes inflammation and itching. But in females, it may actually not be it may not be registered as more than a nuisance, and it might be attributed to all sorts of other things like yeast infections and ignored, and which may increase its propensity for spreading through the rest of the reproductive tract. And obviously, anybody who is infected with gonorrhea is infectious. So in females, that hidden infection can be rather dangerous. In males, it causes a very soon after infection, it will cause a urethritis, an extremely painful urination, which usually will send the person to the doctor. If they do not go, then very often there is pussy discharge from the penis, which is called gleat. And if it's ignored, or if it's improperly or inadequately treated, it can spread through the rest of the reproductive tract, as I explained. But gonorrhea is also, depending on people's sexual practices, gonorrhea is also recorded as a pharyngeal infection in the throat, and anal gonorrhea is extremely common as well. And it is a cause of infertility because of its propensity for infecting the rest of the reproductive tract. So the other species to pay attention to is Niaceria Meningitis, and Niaceria Meningitis is the causative agent of meningococcal meningitis. Now, this is so there are some mysteries around this situation because Neisseria Meningitis is commonly recorded as a part of the normal nasal flora of human beings. It's I would say nasopharyngeal flora. We have a rich microflora throughout our mouths and throats and noses, and Neisseria meningitis is one of those bacteria. But for reasons which are not entirely clear, a situation can arise where, rather like Niaceria Gonorrhea, it begins to infect the cells of the respiratory tract as a pathogen, not as a harmless symbiont, but as an actual pathogen, and begins to move, as Neisseria Gonorrhea does, between cells, and passes into it invades the epithelium and passes into the bloodstream, and is particularly dangerous when that then gets carried to the nervous system. And it will initially infect the cerebrospinal fluid, and then the tissues, the membrane surrounding the brain, and especially the meninges. And this is a very, very dangerous meningitis. There are other infections of the meninges, which can occur, caused by viruses, which are not nearly as dangerous. They are more of a nuisance, self-limiting infections, but this one is not. It's an extremely dangerous disease. There is, however, a vaccine, which you can have even as an adult to stave it off. Okay, so let's pass on now to the next really big group. And this is a group which contains many of the bacteria which are probably most familiar to you, and certainly those which are extreme importance clinically and environmentally as well. But you're going to hear many of the sorts of bacteria that you've encountered in lab lie in this group, the gamma proteobacteria. The first one that I'll mention are called, there's a group called the Thiotrichaeles, and this one I'm going to talk about again because it is representative of this really important group, the chemoautotrophs. And this is called Beggiatoa. And Beggiatoa is a colonial bacterium. It exists in colonies of which form filaments like this. And they can be extremely long, and they wrap around each other, and so acquire a certain mechanical strength. They are an aquatic bacteria, and they can be so abundant that these filaments become easily visible, draping the substrate. And they occur very often right above a totally anaerobic sediment, and where the anaerobic sediment meets the aerobic environment above. So they are not anaerobes themselves. They exist, but they live in that zone where the substrate, for example, completely lacks oxygen and then encounters maybe the water above, which is oxygenated. The reason for this is they are aerobic, but they need to use this, again, this is H2S. This is their source of energy. And H2S is produced in abundance in anaerobic sediments. It is the byproduct of breakdown of proteinaceous matter, of the decay of proteinaceous matter in the substrate. And it's a gas, and it's dissolved in water and is released from those sediments, and is utilized by Beggiatoa because Beggiatoa is a chemoautotroph. It fixes carbon dioxide, takes carbon dioxide and water, and again, in a similar process to what we saw for the previous example, it takes the hydrogen and uses it as a source of reducing power. But now it actually leaves the sulfur on its own as elemental sulfur. And you can actually see, if you look here in these threads here, you can see these bright granules here embedded in the cytoplasm of these cells. Those are actually sulfur granules. And Beggiatoa is one of the organisms which is responsible for actually, over geological time, building up large masses of elemental sulfur. The action of organisms like Beggiatoa and organisms like Beggiatoa can end up building up sediments that are full of sulfur, elemental sulfur. Okay, the next one is a disease that you may well have heard of, and this is tularemia. It is caused by this gamma proteobacterium called Francisella tularensis. It is named for the area where it was first recorded, which is Tulare County in California. And it was most commonly associated with infections caused by contact with rabbits. And there were two ways in which that could be, or I'd say more than two ways. Contact with rabbits can be because a rabbit bites, for example. So it could arise from a rabbit bite if people were hunting, grabbed a rabbit or whatever. It can also arise from bites from fleas or that are on the rabbit. And it can also arise from eating rabbit. So that's why rabbits became particularly commonly associated with it. But in actual fact, Francisella can infect many different rodents. And it can be passed into the populations of predators, rather, on those rodents, like ticks and mosquitoes, as well as fleas. And Francisella can be transmitted by any of these as a vector. So when it does, it has quite a complex life cycle, which I won't go into, but it is actually ingested by macrophages and transported around the body, and especially to the lymph nodes, and causes a systemic infection. Here is, you can see it's a very strange structure. It's not quite a rod or a coccus or anything like it. We say that it is pleomorphic. And this is Francisella, which has been ingested by a macrophage. And it has started to breed inside the macrophage. It survives, normally phagocytosis by the macrophage ends up in digesting whatever it has ingested. But there are organisms which have adapted so that they are resistant to that digestion, and Francisella is one of them. Initially, it will cause, when it arises, for instance, through an insect bite or an animal bite, it can cause an distinctive ulceration like this, but then can spread systemically. Very often it is self-limiting, but it can be life-threatening. So it just depends probably on the combination of the virulence of the bacterium and the susceptibility, the genetic susceptibility of the host. There have been instances where Francisella has been inhaled, however. And when it is inhaled, it has a very high mortality rate. It is a rapidly developing respiratory disease, which is very frequently fatal. But fortunately, the instances of it being inhaled are very rare. It's only under specialized circumstances, usually from agricultural dust and stuff that it is acquired, or especially where there's rodent feces and things like that present. So that is Francisella tularensis or tularemia, causing the disease of tularemia, also known as rabbit fever. So this next organism you will surely have heard of in lab. And this is Pseudomonas in a group called the Pseudomonadales, which are named for Pseudomonas. They all have rather similar characteristics. Pseudomonas is an obligate aerobe, and it's very, very common. There are a number of species, and they are common in soils and water that can be widely isolated. They may sometimes occur as part of our normal gut microflora. They have been recovered from the gut microflora, but I have no idea how common that is. I don't think it is very common. Excuse me. No, their main interest, apart from their environmental importance, is the fact that they are also opportunistic pathogens, and they arise most commonly as nosocomial infections, especially in people who are immune compromised or the elderly, and they are absolutely dreaded in burn wards, where they colonize the surface of the skin when it has been badly burned. And there's one in particular that is associated with burns, and that is Pseudomonas fluorescens. The Pseudomonas is very, very diverse metabolically. They're all aerobic, strict aerobes, but they can survive under anaerobic conditions because they produce both nitrate and nitrite reductase. Now think back, we talked about this earlier, when we talked about respiration. We mentioned anaerobic respiration. Organisms which can perform anaerobic respiration are able to take nitrate and use some of the oxygen as their terminal electron acceptor in respiration. So they take nitrate and they reduce it to nitrite. And nitrate is NO3, take an oxygen off, and it ends up being NO2. But Pseudomonas can go further. It can actually utilize all of the oxygens on nitrate, goes from nitrate to nitrite, and then takes off those two oxygens, and it produces elemental nitrogen. So it can produce nitrogen gas and utilize all of the oxygens that were present on the nitrate as terminal electron acceptors, so that it can continue cellular respiration under anaerobic conditions. Another species, which Pseudomonas aeruginosa, also a very common environmental bacterium, is known for infecting wounds, and especially for setting up urinary tract infections. These can be quite difficult to treat and get rid of. The urinary tract infections are often through the use of catheters in healthcare settings, but they can arise spontaneously as well. So here it is. This is Pseudomonas, polarly flagellated, but usually only one end, not like Spirulinum, which had at both ends. And it's a distinct rod-shaped gram-negative bacterium. Okay, so here is another one, again, of particular interest in health, but actually and in widespread environmental bacterium, and this is called Legionella. There are a lot of species of Legionella. A number of them can cause disease, but this one, Legionella pneumophila, causes the most serious of all of these diseases, which are all respiratory diseases. Now, Legionella is a very common aquatic bacterium, and it occurs even in very small bodies of water. Legionella can easily be isolated, especially when it exists together with a number of other organisms, especially some amoebae, algae, and things like that. So it's part of what we refer to as a consortium. That is a lot of different organisms that interact with one another and grow best when they are in association with one another. So Legionella grows best, or achieves high population numbers, let's say that, in warm, still water with some exposure to light, where other organisms like the amoebae and everything are existing. So that is fine. That's easy to understand in environmental terms. But unfortunately, that is also this kind of situation that arises completely artificially in air conditioner systems, for example. In many of our machines and things which are cooled, there are drip trays and things like that, where water can accumulate and where Legionella can breed. Now, Legionella pneumophila causes a disease called Legionellosis, which is a dangerous pneumonia. And it was named for the fact that it was first recorded at a meeting of the American legionaries, the veterans, at a convention in Philadelphia. And they met in a convention hall, and it was summer. The air conditioners were running, and then a large number of these rather elderly gentlemen went down with a very virulent pneumonia, and a lot of them, I believe about 30, actually died as a result of this epidemic. And it was a great mystery. Nobody knew why on earth all of a sudden all of these people who had nothing else to do with one another all went down with the same disease. It was obvious there was some common source, and it took some considerable amount of investigation to discover that the air conditioning system was sitting idle for a while, and then in the drip trays, this organism was breeding up, and then when the air conditioner switched on again, they disturbed the water in the drip trays and caught up droplets of water, spread them through the air conditioning system, and sprayed them out into the room where these men were meeting. And it has since been discovered that, in fact, this has happened a number of times. And it is usually associated with these heating and cooling systems. Nowadays, a lot of attention is paid to isolating the drip trays and things like this from the rest of the machine, so that this doesn't occur. Modern air conditioners don't do this. They use different systems. But nonetheless, this still occurs. And in fact, I think about 3 years ago, I can't quite remember, but about 3 years ago in the Bronx, there was an outbreak of Legionellosis. And it was very interesting because it was actually people who had been walking on the street outside a particular old apartment building, which had a very aged air conditioning system up on the roof. And this air conditioning system was creating an aerosol, and that aerosol contained Legionella and was inhaled by the people passing along the street. So it causes a typical respiratory, lower respiratory tract infection and a pneumonia. And it does have a fairly high mortality rate, especially in the elderly. But there are milder forms of it. It's often called Pontiac fever. And that tends to occur in younger people infected with Legionella. Okay, so the next one of the gamma proteobacteria is a group called the Vibrionales. Excuse me. These are often just called the Vibrios. There are a lot of members of this group, the Vibrionales, but they are all aquatic. And many of them are marine or associated with brackish water, that is water which is a low salt content but has a distinct taste of salt. And the most important one of all is called Vibrio Cholerae. There are, as I say, there's a number of other species. One called Vibrio parahemolyticus causes virulent gastroenteritis, but not nearly as serious as Vibrio Cholerae. There are other Vibrios, especially marine ones, which are associated with that terrible thing called the flesh-eating syndrome. The flesh-eating bacteria is usually not just a single bacterium. It's usually a collection of bacteria that acts synergistically to set up those terrible conditions where the connective tissue is digested away. And some of the Vibrios have been associated with that. So they occur environmentally. They occur widely as completely free-living organisms, but their ingestion or their association with humans can set up very dangerous disease situation. Vibrio cholerae is the causative agent of the disease of cholera. And cholera used to be regarded as being a plague organism. It used to run rampant through crowded settings, particularly where there was poor sanitation, for reasons that will become obvious. Now, Vibrio cholerae can be isolated from aquatic systems very widely. It tends to be more commonly associated with brackish water, but it can also be isolated from fresh water. And when it is ingested by a human being, it causes a virulent gastroenteritis and a violent diarrhea. In fact, it releases toxins. It releases exotoxins, which cause the cells of the intestine to flood out fluid. So the cells throughout the intestine pour out fluid, and they, to such an extent that there is a very distinctive stool which is associated with cholera, and it's called a rice water stool, because very soon the entire gut is flushed out, and all that is passed is a thin fluid, which is a slightly gray, pearly look to it. It's basically body fluid. People actually die of dehydration, of hypovolemic shock, that is loss of blood volume, and because all the fluids just flood out in the form of diarrhea. And cholera used to have a very, very high mortality rate, which is why it spread very easily, because once it entered a population, if that population lived as people used to do routinely in early times, if they lived with poor sanitation, especially where things like pit latrines were able to, were accessing the water source, then the bacterium would spread very widely, very quickly, because it would begin to contaminate water sources. Once it contaminated water sources, of course, it spread widely through the population, and even more would enter the sewer system. Cholera still occurs quite widely here, and it's been discovered that, in fact, again, it is part of a normal ecological association. And here's its most important. It is with these small crustaceans called the copepods, which are incredibly common in fresh and marine and in brack waters. These copepods are very, very common. And Vibrio cholerae has a propensity for settling around the mouth parts, especially. Here's obviously the crustacea, they have an exoskeleton of chitin. Here's a picture of Vibrio cholerae cells attached to the exoskeleton. And these organisms periodically breed very, very rapidly and bloom, especially in spring and in autumn. They bloom in the water source, and along with them blooms Vibrio cholerae. And people drinking this water will inadvertently drink both free Vibrio and huge concentrations of Vibrio attached to these little animals. So this has led to one very simple public health measure. Cholera still occurs very widely throughout the world. It's endemic in India, and especially up in Bangladesh, these areas up here, Pakistan, Bangladesh, around here. That is widely regarded as being the source, if you like, of most of the epidemics that have passed through Europe and Asia. But it occurs very, very widely in Asia, in Africa, in Central America, and then, very surprising, not shown on this map, but there is a large number of cases have been recorded, relatively large number of cases have been recorded from Canada, is not clear, at least not clear to me, exactly why that should be. But it raises its ugly head all over the place with regularity. It's often little spotty outbreaks are recorded even in North America. But it is a very distinctive disease. And people, when they start feeling ill, they very quickly go to the doctor. And it is treatable with supportive therapy by intravenous fluid replacement, for example, but also with antibiotics. But in fact, even in the absence of antibiotics, the disease can be successfully dealt with, provided people are hydrated properly. If they are hydrated properly, then it becomes self-limiting. They eventually flush it out completely. Nonetheless, it's a very dangerous disease, and it produces a very high mortality, especially under epidemic conditions. One of the reasons for that is that those epidemic conditions often arise under disaster situations, after earthquakes, floods, these kinds of things, when sewer systems are disrupted, and when people themselves are disrupted, when their movement and everything is disrupted. Then cholera tends to become rather more common. In the absence of antibiotics, people can survive cholera provided even with the simple household remedies. There are recipes for fluids which they can drink, containing sugar and salt, for example. There is a vaccine. The vaccine is not a particularly good one, but it is good enough. It is a relatively short-acting vaccine, but it's good enough under epidemic conditions to protect many people, at least for long enough until the epidemic passes. But proper sanitation is the key to the limitation of cholera as a disease-causing organism. Okay, so I'm end with that, and then I will hopefully, provided I'm well enough, I will see you tomorrow. Okay.